Hello and welcome to El Zoominario. We have today Fabricia Nascimento joining us to talk about a framework to simulate HIV epidemics. Fabricia was born in Rio de Janeiro in Brazil. She has a bachelor degree in microbiology and immunology from the Federal University of Rio de Janeiro and a master's degree in molecular and cell biology from Oswaldo Cruz Institute, also in Rio de Janeiro. And during this time, she was working on population genetics of howler monkeys. She has a PhD in evolutionary biology from the University of Sydney, where she was working on the evolution of endogenous retroviruses. She's currently a research associate at Imperial College London, working on molecular epidemiology of HIV and other viruses. Thank you, Fabricia, for being here.
Oh, thank you, Claudia, for inviting me. So I'm Fabricia, and I'm a research associate at Imperial College, and I'm working with Eric Voss. And as a collaborator in this project, we also have Sanjay Mehta and Susan Little from University of California, San Diego. And today I'm going to talk about a framework that I have been developing to simulate HIV epidemics. So before I start with my work, I'm going to give a little bit of background. So HIV is a virus that infects millions of people in the world. And there are millions of infections, like new infections per year. So imagine a population. So here represented by these black circles. And imagine that in this population, you have some infected individuals here in red. And these individuals here, they don't receive any form of treatment. Some other individuals here, like in yellow, they are HIV positive and they receive treatment. But there are many other individuals here represented in green that they are HIV positive, but they don't know that they have not been diagnosed yet. So now imagine we get one of these individuals and he's going to get tested to see if he's HIV positive or not. So he provides a sample, and often this person can also allow that their virus can be sequenced. So you have the genetic sequence of the virus. And some people, they're going to have access to all the information related to this virus, such as their address, their name, the date of birth, and any other information associated to this patient. Imagine now that many individuals like that allows their virus to be sequenced, then you can get, you can try to understand how these different viruses are related. And for that, we use what we call phylogenetic tree, which is this schematic illustration here. And we use this tree to understand how these different viruses, they are related to each other. So now imagine that some individual, like a researcher or maybe a doctor, they have access to more information than just the sequence of the virus, such as the name of the individual, the address, their date of birth, and you can have this for all of these individuals. So this project came to try to understand the risks and the benefits of having these HIV genetic sequences. So for us to try to access that, we have to develop a very simple framework to simulate the conditions to understand the risks and benefits. So then I developed this R package called HIV EPSYN. So R is computer language, is a statistical computer language that you can use to generate graphs and also do simulations. And this package is also based on another package called EPI model. So EPI model allows a person to simulate an epidemic on a network. So what is a network? So here you have a GIF illustrating a network, and you have in blue susceptible individuals, and you have in red infected individuals. So it's interesting to know that doesn't need to be individuals. So if you're interested in understand the animals, so your node here is an animal. in my case, I'm interested in individuals, and I'm simulated men that have sex with other men. So here you also can see that some of these nodes, they created this edge with another node. That means they create a relationship. And when a susceptible individual is in a relationship with an infected individual, the susceptible individual can get infected. So epi model has three main components. So one of them is the elements of your network. So in my case, these nodes are individuals, as I mentioned before. But you also have the states, which are the attributes of this individual. So this means that you have to define the age of this individual, if this individual has been tested or not, if they are susceptible or infected, if they are on treatment or not. And the third component is the transitions. So you have also to specify how things happen in this network. So how an individual dies, how an individual arrives in this network, and so on. So why we chose an agent-based simulator? Because in an agent-based simulator, the individuals, like the nodes of your network, they are uniquely identifiable. And the individuals are tracked over time. So I'm able to know who infected whom and what time that infection happened. And I know any other metadata that I wanted to know. And also, EpiModel allows you to create another package. That's what I did. And it has this modular approach. So this means I have different modules to deal with different situations in the network, such as I need to initialize all the individuals of my network. So I need to tell how many susceptible individuals I have there at the beginning, how many are infected. For the infected individuals, I have to tell if they are on treatment, if they have been diagnosed or not. So I have to start the attributes of all the individuals in the network. And I also need to define how individuals arrive, which age they arrive in the network, how they die, and also all the HIV-related parameters, such as how they get tested, how they get treated, how HIV progress, because there is the disease progression. So you can start an early HIV stage, and then you can move to a chronic stage, and then and the chronic can be divided more or less 3 different stages, depending on the CD4 count of the patient. And also you have to define how transmission is going to happen. And we are also interested in migration because we want to understand importation rate to a population. So I'm going to explain a little bit about transmission rate and how we did it. So the parameters we chose was based on another publication by Levu and collaborators that has been published in Epidemics. So 80% of the individuals in a network, they belong to a risk group 1, and 20% they belong to group risk 2, which has a 10 times fold of transmitting the disease. Also, an individual at early HIV stage, they are more likely to transmit the disease than an individual at ages stage. And the same for care. So an individual that has not been diagnosed, but he is HIV positive, they are more likely to transmit than an individual that has been diagnosed and is on treatment. And the reason is because once you know your status, you change your behavior. And once you change your behavior, you're less likely to transmit as well. Also, when you're on treatment, the treatment also decrease a lot your chance of transmitting the disease. And the thing we're implementing that is new is migration. So I create a way that you have 2 different populations, a population 1 that belongs to region, because it's the region I'm interested in analyzing. In the case of our project is San Diego. And they have a bigger population, that is population 2, and this is what we call global population. So in the way we build the model, we allowed people from the same population to form a relationship, but we don't allow a person from population 2 to form a relationship with a person in population 1. So pretty much we don't allow traveling here. So if someone has a boyfriend in another country, that is not a counted in our model. And a person can migrate to another population, and once that person migrates, that person becomes part of that other population. So a person population 2 migrated to population 1, and then this person becomes part of population 1. So the outputs of my model, I have epidemiological quantities of interest. So here's just an example. So here's a graph that's in the x-axis here. I have the time in units of days. And in the y-axis here, I have the number of individuals. So here's just for population 1. So in red, I have the number of susceptible individuals that decline with time. And in blue, it's the number of infected individuals that increase with time. And the second output I have is a transmission matrix. So in a transmission matrix, I have the time an infection happened and who transmitted to whom. So I know who was the infected person and who was the person that this infected person transmitted to. So once I have this transmission matrix, I can convert to a phylogenetic tree that I mentioned at the very beginning of the presentation. And then I know the relationship of all these tips of this phylogenetic tree. So this is, I call the true tree, but quite like almost all the time, we don't know this. We don't have this information. We don't know the truth. We don't know who infected who in most of the cases. So we also are interested in getting the genetic sequence that could generate this tree. And then we try to estimate this tree again and see how the results compares like. And why we are doing all of these? Because we are trying to characterize the risks from collecting this sequence data. And to do that, I'm using statistics that's called the infector probability that has been described by Voss and Frost in 2013. So this is the probability that a given person is the source of infection for another person. And for using this statistic, I need a phylogenetic tree, which I use the genetic sequence of the virus, but I also need surveillance data. So surveillance data in this case would be the total number of people living with HIV in that specific region that I'm interested. I also need the number of new HIV infections per year. And I also need to know which HIV stage of infection each individual is. And by using this infector probability, I'm going to try to quantify the risk of disclosure and identifiability of transmission pairs. And the reason is because when you provide a sample, some people are concerned that they can be identified or they can know who they transmit or who transmitted the virus to them. So this is to inform individuals. And sometimes what's important to note, many of these people, they don't want the information to be disclosed. They don't want their family to know they are HIV positive. Sometimes when they are MSMs, in some parts of the world it's illegal to be MSMs, is also a crime. And many other personal reasons, they don't want this information to be disclosed. So I did some very preliminary simulations. So I simulate a very small population. So my population 1 has only 1,000 individuals. And from these 1,000 individuals, 4 are infected. And my population 2 has 2,000 individuals, which 10 are infected. So I simulated in days, the equivalent of 40 years. And after the first initial 24 years in days, an individual allowed to be offered treatment. And I varied two different parameters in my model, the transmission rate and the migration rate. So here is a plot that I trying to understand the infector probability. So the columns represent the different migration rates. So the last column here, simulation 4 and simulation 8, I don't have any sort of migration. And in column 1, the migration rate from population 1 to population 2 is the same as from population 2 to population 1. And it's the second column, the migration rate from population 1 is smaller than the other way around. And in the third column, I have migration rate from population 1 is higher than from population 2 to 1. And the different rows, I have the two different transmission rates that I analyzed. So in the first row, I have a higher transmission rate, and in the last row, I have a lower one. So what I'm plotting here, so I got infector probability that was higher than 80%. So I would say that I have a higher confidence that that transmission pair is actually a true transmission pair. And I plot all the combinations of pairs that had an infector probability higher than 80%, which is this kind of red line here. And then I check, because remember, I know the truth because I'm doing simulation. So I check all these pairs to see which actually was a true transmission pair in which I correctly identified donor and recipient. And this is the line in green, was very close to the red line. And in blue is when I got the transmission pair, but I incorrectly identified who was the donor and who was the recipient. And they are very similar. The only problem here is that I only did 20 simulations because this is still in a test stage. So I'm still trying to see if my code is working correctly or not. So I still need to run more replicates. The other thing we did is what we call a ROC curve. So this has this receiver operating characteristic curve, and you can plot. So you plotted this curve to understand how well the infector probability is actually detecting a true transmission pairs. How well is this classification happening? So in the case for this, I only check infector probability for what we call a cherry. So a cherry, you have a tree, and it only composed by, let's say, 2 individuals of that tree when they group together. And then I check how well they classify, like the infector probability performed. So here, ROC is important to know. So that ROC in the x-axis, I plot the false positive rate, and the y-axis, I plot the true positive rate. So an amazing classifier, when I'm able to classify everything correctly, I would put my curve very here on the top. So you would minimize your false positives, and your true positive was going to be 1. So you can see my curve go really down here for all of them. And then I calculate the area. So the area under the curve would be all this area here. And on average, let's say like this was around 75%, which means that I had a 75% chance of correctly identifying a transmission pair. So I plan now to simulate bigger populations because 1,000 individuals and 2,000 individuals is not enough. And then I plan to compare the truth, because I know everything, with the estimated. That's actually what we have in science. We have only the sequence, and we estimate everything from there. So I want to know how good I can estimate transmission pairs using the estimated trees. And I'm also gonna use phylogenetic analysis. So phylogenetic analysis that you can use to try to use a phylogenetic tree and estimate parameters such as the transmission rate and migration rates. And that is another step of our project. And in that way, we plan to quantify risks and benefits to have all this genetic sequence, HIV sequence available. And some benefits we plan to simulate is to simulate interventions. For example, what happens if we increase the test and treatment rates? So it's very important to notice that we have a public database of genetic sequence, DNA sequence, all available. But for HIV and many other maybe diseases, you don't have, so you can get the genetic sequence, but you don't know who that sequence belongs to. So you don't have their name there, you don't have their address, you don't have any of this information. In many cases, you do have the location that it came from, but the location is very broad. It's not gonna give you a postcode there. It's gonna have like, oh, this sequence come from the UK, for example. So this is more to inform people, and they may be afraid that once they provide the sequence, that all the information is going to be available to everyone. So I would like to acknowledge the funding, which comes from the NIH, and the collaborates on this project from the Johns Hopkins and the University of California, San Diego, and Emory University. So that's the part of my talk. And so I've been asked to talk about something about my culture. So I'm from Brazil. And I decided to talk about New Year's Eve in Copacabana Beach. So probably what pretty much everybody knows is that it's a very big celebration, well known around the world. And it brings millions of people together in Copacabana. So here you can see the fireworks and to see live music. So it's a very big celebration. What people might not know is that we have a lot of rituals during New Year's Eve. So people come to the beach to give offering to Iemanjá. So Iemanjá is a orixá, so let's say it's a sort of spirit, and she's also known as the goddess of the sea. So the orixá comes from religions like the Umbanda or Candomblé. So there are two different religions that originated in Brazil. So what happens, so that when the slaves arrived in Brazil, they were forced to be Catholic, but they had their own religion and their own beliefs. So they end up joining, and it creates these religions based on syncretism. So they kind of was associated these orishas to the Catholic saints in a way of having still be able to have their own beliefs. So there are many other orishas, but in New Year's Eve we make offerings to Iemanjá. And here is a picture of people from Umbanda, and they are celebrating as well at the beach. And you can also make offerings such as light candles and putting your champagne at the beach and, I don't know, pray and make your wishes. So Iemanjá is also the mother of all of us. So she's equivalent to the mother of God, let's say, in Catholicism, which now I forgot the name in English. So we also eat pomegranates. So the tradition says you should eat 7, and then you keep the seeds in your wallet to bring good fortune for the year. The same with the grapes. You should eat 7 as well. And you can also keep 7 seeds if you wish to also bring good fortune. It's very common to eat lentils with the same purpose. Like we want to bring all the good fortune of the world, I think. But you should not eat poultry. And according to my mom, they say that chickens, they scratch backwards and you want your life to move forward. So you should not eat chicken on the New Year's Eve. And there's also the tradition of the underwear. So you should choose the color that you like for your life in the new year. So for example, if you choose pink, then you're gonna have love for the new year. So the most popular color is yellow. So you buy your yellow underwear to bring money and good fortune. So you can also dress all yourself in the color if you want as well, but it's more common that people are gonna dress in white. So here is myself at the beach. So also when the years change, as soon as it's changed, so everybody goes to the water. You can see lots of people here to jump the 7 waves. So for each wave, you make a wish for the new year that is coming. So here is myself at the I think that's Ipanema in 2013. Also, I am baptized and I don't have a religion. And I'm also making my offering to Iemanjá. So we can offer roses or this other type of flower, which I think is gladiolus, so something like this in English. And you make an offering, you make a wish. And like me, it's many people in Brazil at least, so in Rio at least, they have their own religion that is not Umbanda or Candomblé, but they still make the offering to Iemanjá because, yeah, if it doesn't hurt, so why not? So if you decide to spend the New Year's in Rio, make sure you have your new white clothes, plus choose your color wisely for your New Year's to bring good fortune to your New Year's. Thank you. So I'm happy to take any questions.